Drug development is a complex, time-consuming and cost-intensive process. It requires the physiochemical determination and evaluation of a new substance. Furthermore, bioavailability, bioequivalence, as well as toxicological aspects have to be examined. To gain approval for any pharmaceutical drug, the claimed effect of the intended target site has to be proven. Two parameters are used to describe the absorption and distribution as well as the biochemical and physiological effects of a drug: pharmacokinetics, PK, and pharmacodynamics, PD. The gold standard to determine PK and PD is plasma concentration, independent of how the drug is applied to the body and independent of where the drug should establish its effect. It is, however, questionable whether plasma concentrations reflect concentrations at the target site, for example, in the skin or the brain. Therefore, the FDA has indicated a need for tools to assess drugs and their effects directly at the target site, rather than indirectly via plasma concentrations. To meet this need, our researchers from the Institute of Biomedicine and Health Sciences at JOANNEUM Research have developed a unique tool, the OFM technology. A minimally invasive catheter is inserted into the target tissue. Its grid-like structure enables direct contact with the interstitium. Interstitial fluids and biomarkers can be collected continuously and used for simultaneous analysis of multiple substances, independent of their molecular size, lipophilicity, and protein binding. By using OFM, PK and PD of a drug can be measured continuously for more than 24 hours at the target site. One main field of application for the OFM technology is dermatology. Systemically as well as topically, applied drugs can be analyzed for their target tissue PK and PD. In this schematic illustration, the OFM catheter is placed in the dermal layer of the skin. A topical drug is applied to the skin, penetrates its corneal layer and diffuses into the epidermis and dermis to exert its therapeutic effect. At the same time, the drug, biomarkers and interstitial fluid are collected in the OFM catheter. In contrast to blood samples or biopsies, our minimally invasive probe allows repeated sampling of the interstitial fluid for PK and PD analysis. OFM also allows sampling of drugs that are bound to a protein or an antibody. By using OFM and gaining direct in vivo access, all endogenous entities and xenobiotics of interest are accessible for subsequent analysis with our highly specific analytical methods. In our clinical studies, several OFM catheters are used simultaneously to assess lesional as well as non-lesional sites in a single subject, reducing inter-subject variability and raising statistical power. This reduces the required number of study participants by a factor 10. Due to the multi-catheter setup, we can collect enough data on drug effectiveness as early as a clinical phase 1 study. This can reduce the risk of redesigning a drug after a clinical phase 3 study, where hundreds of study participants have already been assessed. Costs can be saved because of the pharmacological profile of a drug is known at an earlier phase in drug development. The OFM technology has proven to be a unique, safe and valuable tool for preclinical and clinical PK and PD studies. This tissue-specific method is minimally invasive and continuously samples interstitial fluid in vivo. It provides a complete biochemical picture directly from the site of action. OFM can be used at an early stage in drug development and opens up new horizons in preclinical and clinical drug testing. JOANNEUM Research, together with the Medical University of Graz, is your competent and qualified partner for preclinical and clinical studies using the OFM technology. We use the OFM technology to perform PK and PD studies, to investigate bioavailability and bioequivalence, and to perform metabolomics in vivo and ex vivo. We can assist you in preparing the study protocol, submitting the ethical application, executing preclinical and clinical studies, validating the necessary bioanalytical procedures, and statistically analyzing the data. We look forward to help you understanding what's going on with your drug at the target site.